Welcome back, everyone. We are discussing our cytoskeleton. We just discussed our microtubules, and now we're going to start exploring our microfilaments. Microfilaments are the smallest of the cytoskeleton filaments. Their most important role is muscle contraction, probably their best described role in muscle contraction, although they're also involved in other types of movement, so cell migration, allowing a cell to move, amoeboid movement, that kind of, if you've ever watched an amoeba kind of do a little modern dance kind of thing, and cytoplasmic streaming, which is the movement of cytoplasm and its contents within a cell. Microfilaments also help maintain and develop the shape of a cell because those microfilaments are attached right underneath the plasma membrane. And they're also the core of microvilli. Microfilaments are made of a protein called actin. Actin is very abundant in all eukaryotic cells. And when it's synthesized, it folds up into a globular shaped monomer, so a little round shape. It combined either ATP or ADP. G-actin is globular actin, and G-actin can polymerize to form microfilaments of F-actin. Actin is really highly conserved through all different types of cells, although there are a little bit of variations here. So we can kind of broadly divide our actins into the alpha actins, which are the muscle-specific actins, and the beta and gamma actins, which are the non-muscle actins. And the beta and gamma actins will be found in different regions within the cell. Microfilaments are polar. They do have different ends at their different ends. And this is shown by the different way that G-actin will be added or lost more rapidly from the plus end than it is from the minus end. And after the G-actin will assemble onto the microfilament, the ATP is going to be slowly hydrolyzed. So the growing microfilament ends will have ATP actin because that slower hydrolysis, the ATP is kind of backing up there. At the other end, most of the microfilament is composed of ADP actin, where that ATP has already been hydrolyzed. There are drugs that can affect the polymerization of microfilaments. One of them will be cytokinesins, one of them will be the trunculin A, and one will be phalloidin. So cytokinesins are fungal metabolites, so you find them, you would be exposed to them by eating these fungi. They prevent the addition of new monomers to existing microfilaments, which means that your filaments will depolymerize. Trunculin A is a toxin that sequesters actin monomers, prevents them from being added to microfilaments, and phalloidin will stabilize microfilaments and prevent them from being depolymerized. So someone who has been eating toxic mushrooms, these are some of the metabolites that they would have been exposed to. Cells can take their actin and dynamically, actively assemble it into different structures. So cells that are adhering tightly to their underlying layer are going to organize their actin into stress fibers. A cell that's rapidly moving isn't going to have these stress fibers, are not adhering as tightly, they're skimming, so they have differently shaped actin structures. And then the cell cortex beneath the plasma membrane has actin cross-linked into a gel. Cells that are crawling along a surface are going to have lamellipodia and filopodia at their leading ends. It kind of reaches out and grabs onto the substrate and pulls them forward. These lamellipodia have this branched actin kind of fills in and allows the cell to move in this direction. So the filopodia are here at the leading end. Microfilaments are going to be really highly oriented. The plus ends are going to be moving and forming new microfilaments in this way. Cells are able to control where actin is being assembled and able to control the structure of the actin network using different actin binding proteins. They can control during nucleation, during elongation, and during severing of the microfilaments, and by associating the microfilaments into different networks. This allows a cell to control how actin is being polymerized, how long it is, and how organized or branched it is. Whether microfilaments are able to grow will depend on whether their filament ends are capped. So capping proteins are proteins that can bind to a filament, and by the presence of that capping filament, that subunit is going to be a little bit more stuck in its place. It's going to prevent the loss or the addition of subunits there. One capping unit is cap Z, which binds to the plus end of our microfilaments, and it prevents adding additional subunits there. Tropomodulins bind to the minus end and prevent the loss of subunits from the minus end. Actin networks can form as these loose filaments, loosely cross-linked filaments. One of the proteins that's important in helping these cross-link into filaments is called filamin. So filamin is kind of a splicer. It joins these two microfilaments together and allows them to intersect. Microfilaments can be broken up by proteins that sever and cap them. One will be gelsolin that breaks actin microfilaments and caps those exposed ends to prevent further polymerization. And then some actin-containing structures are really highly ordered. So actin can be organized into these tightly organized bundles called focal contacts or focal adhesions. Alpha-actinin is a protein that is really important in creating these focal adhesions. And then fastin is a protein in filopodia that helps keep the actin tightly bundled there. Okay, we're not done with our actin filaments yet. We have a little bit more to discuss. I'll see you back in a little bit. Bye.